Welcome to our YouTube channel, and in today's tutorial I'm going to be explaining glycolysis. Specifically, we'll be looking at the investment stage from step 1 to 5. Now, glycolysis basically means splitting sugar. If you eat carbohydrate food, you're going to metabolize that carbohydrate to produce glucose, which is the simplest sugar that we constantly find in our blood. And glucose is a 6-carbon molecule. Here is glucose. 1, 2, 3, 4, 5, 6. 6-carbon molecule. And it is in your blood vessel. This is artery. Artery supplying the various cells with nutrient and oxygen. So there's your blood cell, and you see oxygen is seated right in the blood cell, and your blood wants to deliver oxygen to your cell, and it also wants to deliver the nutrient, which is glucose, to this cell. Energy is the ability to do work. You cannot do work without energy. Right now, as I'm speaking, I'm using energy to speak. How do I generate the energy? So let's look at it. When the blood reaches here, because blood transport things in our body, it takes this particular glucose and deliver it to this cell. Take for example, we're looking at your muscle cell. Now, glucose comes here. When glucose enters the cell, it has to be phosphorylated. Phosphorylation means we're adding phosphate to a particular molecule. So if this is a glucose molecule and it enters the cell, there is an enzyme that resides in the cytoplasm of the cell that will take one phosphate from ATP. ATP means adenosine triphosphate. Remember, the currency used in Liberia is Liberian dollar. So the currency used in the cell is what we call ATP, adenosine triphosphate. So this guy we call hexokinase, which is an enzyme, takes one of the phosphate from ATP, because ATP has 3 phosphate. That's why we call it triphosphate. Tri means 3. Remember from triangle. So since it has 3 phosphate, we take one of the phosphate and attach it to glucose on carbon number 6. Who does that? Hexokinase. It goes directly to ATP. Say, oh, give me one of your phosphate, please. Let me attach it to glucose on carbon number 6. That is the first step in glycolysis. So when hexokinase take one of the phosphates from ATP and add it to carbon number 6, instead of us having ATP, we now produce ADP, which is adenosine diphosphate. That means 2. Initially, ATP had 3 phosphates, but we borrowed 1. So we now have ADP. That's where the ADP comes from. So if we take the ATP and add it on carbon number 6, remember carbon number 1, 2, 3, 4, 5, 6. On carbon number 6 is where hexokinase add the phosphate group that it took from ATP. Reaction number 1. So we move to reaction number 2, or step 2. What happens in step 2? There is another enzyme in step 2 called phosphohexoisomerase. If this was in the liver, we are going to call this enzyme phosphoglucose isomerase, because only glucose kinase in the liver that catabolize reaction number 1. So in reaction number 2, it would have been phosphoglucose isomerase in the liver. But since we're discussing muscle cell, we have phosphohexoisomerase that catabolize reaction number 2. Now, what the guy can do, it will isomerize glucose 6-phosphate into fructose 6-phosphate. Now, glucose is an aldose sugar. It has aldehyde functional group attached to it. But fructose is a ketose sugar. It got ketone functional group attached to it. But both of them, they are all 6 carbon. That's why phosphohexoisomerase is just to isomerize it. That is same 6 carbon molecule, but it's just changing from aldose sugar to ketose sugar because fructose is a ketose sugar. So this guy phosphate so and some ways will convert glucose 6-phosphate into fructose 6-phosphate. You're just changing it around. Think of it like I'm the same person, but if you turn me upside down, my hair will be down and my foot will be up. Then I still remain in my nose. But this guy, they are the same 6 carbon molecule, but both of them they are isomer. They have the same molecular formula, but the structural formula different. Fructose 6-phosphate in reaction number 3 will be converted to fructose 1,6-base phosphate. Now, remember this reaction we have 1, 2, 3, 4, 5, 6. Carbon number 6, there was a phosphate group attached to it. So we call it fructose 6-phosphate. The reason I changed the color was dealing with glucose. This was glucose 6-phosphate. Because we isomerize it, I changed the color to blue so you can know that this is isomerization. They have the same 6 carbon molecule, but the structural arrangement different. But since we have phosphate group attached on carbon number 6, in reaction number 3, there is a guy they call phosphofructose kinase. For short, we call it PFK1. P for phosphate, F for fructose, and then the K for kinase. What PFK1 normally carry out is that he converts fructose 6-phosphate into fructose 1,6-base phosphate by borrowing another phosphate group from ATP. The same thing kinase did here today is what PFK1 doing. You will go to ATP and take one of the phosphate from ATP and add it now on carbon number 1 instead of carbon number 6 because there is already another phosphate added on carbon number 6. So PFK will take the phosphates from ATP, bring it all the way and add it on carbon number 1. Whenever you borrow one phosphate from there, will now produce another ATP. So PFK convert fructose 6-phosphate into fructose 1,6-base phosphate. Why? Because if you take phosphate and add on carbon number 1, this is carbon number 1 and this is carbon number 6. So we say fructose, because the sugar here are ketose sugar, fructose, 1 for carbon number 1, 6 for carbon number 6 to indicate that there is a base phosphate attached on both of them. We call it base phosphate because the phosphate group they are far away from each other, very far away. And when closer to each other, we are going to call it biphosphate. But this is base phosphate because these two phosphate groups they are separated with carbon molecule, far away from each other. The next reaction, which is reaction number 4, we now have fructose 1,6-base phosphate. The next enzyme called aldolase is the one that catabolizes reaction number 4. Aldolase will come and say, but since these two carbon molecules there is a bond holding them, I want to split them. It will break the bond. You see, it will break the bond. When you break this bond, there where the name glycolysis even come in. Splitting sugar. So aldolase is the one that gave the name glycolysis. Aldolase will come in directly and break the bond that holds the 6 carbon molecule. When you split it, you now have 1, 2, 3 with a phosphate coming one side, 1, 2, 3 with a phosphate coming another side. So you got 3 carbon molecule with a phosphate group attached, and another 3 carbon molecule with a phosphate attached to it. There is a bond here holding them. Now, because aldolase split the fructose 1,6-base phosphate, each of the molecule have different names. This is also a ketone sugar, while this is an aldose sugar. Because it got added have functional group attached to it, while this one get keto or functional group attached to it. Now, the first group on this side is called dihydroxyl acetone phosphate. For short, we call it DHAP. The D for dihydroxyl, the D for di, the H for hydroxyl, The A for acetone and the P for phosphate. The next carbon or 3 carbon molecule on the side with a phosphate attached to it, we call it glycolysis 3 phosphate. Remember on carbon number 3, there is phosphate group attached. 1, 2, 3. There is phosphate group attached. And this entire thing, it has added a functional group attached to it. That's why we call it glycolysis 3 phosphate. For short, we call it G3P. G3P. Reaction number 4 just ended with aldolase. The last step, because we told you in the video, we explained from step 1 to 5. The last step, which is step 5, this dihydroxyl acetone phosphate like to be converted to the same glycolysis 3 phosphate. This reaction is reversible reaction. It means that glycolysis 3 phosphate can be converted to dihydroxyl acetone phosphate, or dihydroxyl acetone phosphate can be converted to glycolysis 3 phosphate. The reaction is reversible reaction. You take this and send it on outside, you take the other one and also convert it to this one. But mostly, in order for glycolysis to go on, dihydroxyl acetone phosphate is the one that can be converted to glycolysis 3 phosphate. Which enzyme catalyzes this reaction? Which enzyme can come to dihydroxyl acetone phosphate and convert it to glycolysis 3 phosphate? We call it enzyme triose phosphate isomerase. It isomerizing the whole thing. The same way you have phosphohexyl isomerase isomerizing glucose 6-phosphate to fructose 6-phosphate. You have triose phosphate isomerase. Triose, because there are 3 carbon molecules for triose. 3. Tri means 3. So you're trying to isomerize dihyd to G3P. That's why I indicated 2 G3P. The original G3P is going first, and the dihyd, which is about to be converted to another G3P. So we have 2 G3P that will follow from step 6 to step 10 in the energy generation stage or the harvesting stage. So these are the 5 steps that we call investment stage. Where it actually came from. In step 1, we're investing. What are we investing? We investing ATP, energy. We borrow phosphorus on ATP and invested it on carbon number 1 when glucose entered the cell. Who did that? Hexokinase. Reaction number 2, we never invested anything. Reaction number 3, phosphofructose took phosphate from ATP and attach it on carbon number 1. We're investing again. So on reaction number 1 and reaction number 3, we invested energy. Think about it that way. But in the energy harvesting stage or generation stage, we're going to tell you how we're generating energy. We invested from step 1 to 5. So watch out in our next video for energy generation stage. Thank you for watching. Don't forget to subscribe to our YouTube channel.